Thank you all. Thank you for the opportunity to speak, and thank you for your care and, most of all, your action. Julie and I have the distinction and pleasure of giving you an update on of some of the work by a Monarch Conservation Science Partnership. This is a collection of scientists and seasoned conservation experts that have come together to try and stem this pattern of reduced population numbers over time. You can see in the graph up on the screen the decline put together in a recent report by the Xerces Society, same sort of information that Wendy was recently showing you. But there's been an 84% decline in the larger eastern population that comes to the eastern U.S. and reproduces, and then a 74% decline in the western population. And so this group was charged with trying to figure out a way to halt that and reverse that extinction. And they did this by putting their minds together, reviewing the science, bringing their own science to the table, producing analyses of large data sets, models, and providing some answers and tools, and basically trying to formulate a conservation strategy, and with that also developing a strategy to monitor the results of some of that conservation that would be put into motion. Wendy covered the life history quite well, but I want to point out a couple things here that it makes it fascinating as a biologist, but also creates some challenges. In the eastern population, the great story is a migrating wave, basically a wave of population production throughout the annual breeding season. And there's the other thing that's important to note is that there is a geographic affinity with each of these generations. And so the challenge becomes, if we want to increase numbers, where is it that we need to provide the resources to do that? And where do we want to focus and where do we want to prioritize? So, some of the recent products of this MCSP, as we call them, the Conservation Partnership, has been to take a look at the extinction risk and develop a target population number. They've also worked with a put together a predictive demographic model to try and focus in which of those generations may be most important or may give you the biggest bang for the buck in terms of increasing numbers. And then they put together a geospatial model that works together with the demographic, some of the information from the demographic model to try and tally where these resources may be, the nectar plants, the milkweed, and this one primarily the first generation focused mainly on milkweed, and figure out where we have those resources, and as time goes on, where we need to put more resources out. And then also there's been some work with the developing some analyses to determine what kind of other threats that there may be affecting the population. So basically these targets and these products provide a foundation for the monitoring strategy and what to monitor from the results, and to also help improve the models over time, and also test some of the assumptions. Now you can see, let me back up. You can see in these arrows here, I have some of the sources. And I will just point out again that a great place to go to get updates from the scientific community is the Monarch Joint Venture site and the Xerces Society, and especially you can get that in a pretty non-technical form, but they'll also give you the citations for the actual work. We have several conservation strategies and targets came out of that work, and Wendy covered those quite well, but essentially we're trying to get 6 hectares for that eastern population, and 6 hectares in the overwintering quality of Mexico, and add up to 1.8 billion additional stems onto the landscape. The work also pointed out that we need to do this a little bit of everywhere, and that we need to potentially, if we're going to meet that numbers, we've got to do some work on croplands. And the objectives of our monitoring strategy are threefold. The monitoring strategy that came from the MCSP was we want to do some broad-scale surveillance-type monitoring, look at status and trends. We eventually want to look at effectiveness of local habitat restoration projects, and we want to engage, we really want to lean on involving citizen scientists in monitoring as well as the conservation projects. We've had several accomplishments so far, and we use that information to determine what attributes to monitor, and Julie is going to show you in the next slide what those attributes are. We produced some methods and written guidance. We tested those methods. We developed a data management system. We will revise those methods. We revised them for 2017. We conducted a formal sampling draw, and this year we actually started collecting some data according to that sampling draw. And I'll let Julie take over from here.
All right. All right. So building on what Pat explained, we selected attributes to monitor in the last, starting in 2016, and these were focused on plant availability, monarch availability, and habitat use, and the site condition itself. We also looked at survival and parasitism by collecting late instar larvae and rearing them in containers and monitoring them to see if any parasitoids popped out, and then we would release the adults. In the Texas region, where the red imported fire ant is found, we also looked at red imported fire ant abundance and density. And the milkweed and the blooming nectar plants, we identified those to species and looked at abundance and density of those. In 2016, we looked at 3 different sites in 3 different U.S. Fish and Wildlife regions, our Mountain Prairie region, our Midwest region, and our Southwest region. And these were on national wildlife refuges or affiliated with them in the surrounding areas. And in 2017, we are also doing a similar study and looking at 3 different locations, 2 in the Midwest region and 1 in the Southwest region, and that is at our Balcones Canyonlands National Wildlife Refuge. We have determined from 2016 work that the time taken to conduct these protocols, the site assessment itself took about 2.5 to 4.5 hours. The adult monarch counts were about 2 hours, usually a little less than 2 hours. And the nectar plant, milkweed, and immature monarchs took 8 hours or less. The people got the hang of that over time. So we have some preliminary results to share with you. These are very preliminary and examples of the kind of data that we can extract from the information we're collecting. So from 2016, this is a very small sample size, but as you can see on the y-axis, we were looking at the number of eggs and larvae, and on the x-axis, we were looking at the sort of land cover types we were finding these within. And on the bottom, the first is the agriculture, in an agricultural fields. The second was CRP lands. The third is roads. The fourth is protected grasslands, and the fifth one, all the way on the right, is unprotected grasslands. And these data suggest that the unprotected grasslands are very important for monarch reproduction, and female monarchs are choosing to lay eggs on milkweeds that are associated in these habitats, and most of these are in private lands. So it really highlights the importance of private lands in Texas and Oklahoma. We have some more results here, again preliminary, but when we looked at nectar plant diversity and where it occurred, we found that roadsides actually provided the highest diversity of blooming plants to adult monarchs, and they were using them along these roadsides. Also, the second most was the protected grasslands in the Midwest and in the mountain region. Mountain Prairie region, and in the Southwest here, we had our our study site was in Oklahoma in 2016 at Washita National Wildlife Refuge. And in that study, unprotected grasslands was a little bit more important than the protected grasslands, just highlighting again the importance of private lands. And it was interesting that this pattern was found in all three of the regions for the preference for the most diversity occurring along roadsides. We also have preliminary data found in nectar plant use by adult monarchs. And as you can see in these two graphs, we look at this in two locations. The Neal Smith National Wildlife Refuge was in Iowa, and again, Washita National Wildlife Refuge in Oklahoma. And the diagonal line divides the amount of use on the top side versus the availability on the bottom side. So when you have elements or plant species in the top part of it, that indicates that they're being preferred more than they are available relative to their availability. And interestingly, in both sites, the monarch adults were preferring a sunflower family species, a Helianthus. In Neal Smith in Iowa, it was Helianthus grossaceratus, and then in Oklahoma, it was Helianthus petiolaris. So in 2017, we employed a more randomized approach where we're using a formal stratified design, which is essentially laying a 10 by 10 kilometer grid across the lower 48 states. And within those grids, each one is assigned a unique number, and those numbers are prioritized and associated with the 5 different strata that we saw at the bottom of the x-axis of the previous slide. And this year, we had a crew of 2 SCA biotech students centered at Balcones Canyonlands National Wildlife Refuge, and they were able to visit actually 6 of the master sample cells in Texas. One of them is not depicted on here. It's at Hagerman National Wildlife Refuge, which is north of this map. But all the squares in yellow are areas where they were able to go and sample. So for monarch monitoring next year, we're very open to that. We encountered some challenges in terms of outreach and coordination and getting the data to the analysts, which were managed on our level, but if this is to expand to a national level, these are things that will have to be addressed, and we've provided hopefully some solutions to remedy these, and one of those would be the Monarch Conservation Science Partnership writing a monitoring implementation plan that would give detailed descriptions of the roles and tasks and time and costs, as well as some power analyses, which we hope to have for you very soon, which will indicate the minimum sample sizes needed. We also are extremely interested in reaching a diversity of audiences and to have agencies and NGOs and academics and everybody, businesses, whoever wants to be involved, basically everybody, to engage in outreach and help us galvanize citizen scientists and promote our monitoring strategy into the future. So if you have questions, we have a handout that indicates all of the science, and it is available in the back table. Thank you.